Welcome to WARF Essential Topics. I'm Eric Iverson, CEO of the Wisconsin Alumni Research Foundation, otherwise known as WARF. In this video, titled Investing in Badger Science to Attract Pharma Partners, WARF Therapeutics, you'll meet WARF's Director of Chemistry, Brian Dyck. Brian will share the background and purpose of WARF Therapeutics, as well as who are the best candidates for this program. WARF Therapeutics partners with UW-Madison and Morgridge Institute researchers in employing an industry-focused approach to improve the value proposition of drug candidates. Thanks for joining WARF Therapeutics. Here's Brian Dyck.
Hello, my name is Brian Dyck. I'm the head of chemistry for WARF Therapeutics. I hope if you're a student or a graduate student or faculty or staff on campus, you've heard of WARF, the Wisconsin Alumni Research Foundation, because we do tend to have a broad positive impact on campus. But what you might not have heard of is WARF Therapeutics, a recent initiative within WARF, which was formed specifically to help biomedical researchers translate their discoveries into the clinic. And so I'd like to spend the next little while introducing you to WARF, explaining what we do, and convincing you that we can help with your projects. So WARF has been around for around 100 years, and they were formed specifically for the purpose of protecting intellectual property that came out of inventions off campus. And so they receive hundreds of disclosures in a year. They file the patents. They protect those patents and manage them throughout their lifetime. Ideally, they realize revenue from licensing those patents. And so they have billions of dollars under management currently. But they were formed independently of the university, but solely for the benefit of the campus. So any money that's recouped from investing that licensing is funneled back into the university in the form of grants, often in excess of $100 million a year. What you might not realize about WARF's war chest is that much of it came from drugs that were discovered right here on campus. The first of those is warfarin, which in the 1940s was sold as a rat poison, but by the 1950s was shown to the FDA and approved there to be an effective blood thinner. And now it's one of the most prescribed medicines in the world. And that work was all facilitated by research right here on campus. And then later in the 1990s, Hector DeLuca discovered a synthetic vitamin D hormone analog called Zemplar, which he licensed to Abbott Labs. And those two inventions really contributed a lot of money to the endowment that WARF now invests. WARF itself is technically independent of the university, but we answer to the same shareholders that are important to the university, including successful UW alumni. And we've formed as a nonprofit exclusively to benefit the campus. So any money that's generated from that war chest gets pumped back into the university in the form of grants, infrastructure, and other initiatives. Recently, people at WARF thought about the potential of targeted research investments into preclinical labs that could potentially accelerate those to drugs, such that we weren't relying on the serendipity to discover badger drugs every 50 years or so. And so to accomplish that, we formed WARF Therapeutics. WARF Therapeutics was formed with an initial investment of $50 million. That was to be pumped into a couple of projects on campus that were independent of disease, independent of modality, but thought were to be close enough to the clinic that a little bit of targeted investment and help from industry experts could drive those to be drugs, and thus formed WARF. So we hired a few people right out of the industry to help. That's myself, Hong Min, as the head of chemistry, biology, respectively, and then our boss, John Young. And of course, we got a lot of support from other people around WARF because there's a lot of experts within WARF itself. We also recruited some of the best people in the industry to help us as a scientific advisory board. These people are experts around in every aspect of drug discovery. And among them, they've put dozens of excellent quality compounds into the clinic. So we really rely on them heavily. We have meetings several times a year, but we call them often. They were responsible for driving all our drug projects forward. However, the most important part of the collaboration is you, we really need professors to come in to complete the partnership. Because by default, we do not initiate any projects de novo. We need a project that a university professor is an expert at to help us, because they are going to be an important component of that project all the way through drug discovery. So where the projects come from is a couple of different sources. Of course, there's hundreds of disclosures a year, and around a third of them are biomedical in nature, so that is one source for projects. But really, the best projects tend to be when we go and meet the professor in their natural environment, show up in their lab or at their department seminars. We knock on their door and say, we've read a paper. Those are where our best projects tend to come from. Regardless of where they come from, though, they're very early in nature as a consequence of academic research. You guys are working on cutting edge biology, of course, but that makes it hard for you to advance to actually finding a clinic-ready compound. And for that reason, it unfortunately limits the licensing opportunities of these projects. This is a plot of what the value of a project, actually it's the value of a company acquired based on its most advanced project. So it's a great surrogate for what a project is worth, because that's how biotech companies are valued. And you can see that a preclinical project just doesn't recoup licensing fees. The reason for that is there's no need for a company to pay a university to license that. Even if a patent is filed, it's hard to protect that intellectual property. That work, when published, erodes some of that IP. So I guarantee that your Nature and Cell and Science and other top-tier journal papers have been read by everybody in the industry. They've loved them, been fascinated, and even followed up probably in their lab. You just never heard about it because there's no need for them to let you know. Everything that they need to follow up is captured in your paper, which is exactly the nature of publishing. But unfortunately, there's no way for the university to recoup money from that. So really, the best way to recoup money from that kind of cutting-edge research is invest some money and drive it to a clinical-ready compound. At that point, intellectual property is easily protected, and we can move forward from there. But it's not easily done in an academic setting. Of course, we realize that. As an example, Hector De Luca made hundreds of vitamin D analogs and tested them. Most professors are unwilling to do that, certainly don't have the funding. And graduate students and postdocs don't find that kind of research particularly rewarding. But we do, and that's why we're here. We can do that work in our labs, drive your excellent science to a candidate, and recoup money for the university and hopefully eventually for you. So ideally, we will drive a project to a clinic-ready candidate. For some premier projects, we have not done this yet, but aspirationally, we would like to drive them through phase 1 into phase 2. And the reason for that is that the UW-Madison campus just has really excellent clinical research groups, and they want to partner with us on these things. So we're just trying to decide on the project that we're going to do that with right now. That's an aspirational goal, but I think that will happen. So I showed you we're a pretty small group, 4 full-time people, and then a lot of other help from WARF, but pretty small, and we don't have any labs on campus. So how do we accomplish this? It's really with what we call our virtual laboratory. We have asked our SAB, worked with them to vet about a dozen really top-notch contract research organizations, or CROs, around the world. And so we are, and in that way, we're not limited geographically. We work in Madison a lot. Of course, we work in professors' labs often. The university has an excellent screening facility, so we can do high-throughput screens there. And even outside campus, there's great companies in Madison we work with. So Immunogen is actually a spin-off from WARF that's great. Promega is a world-renowned protein degrader company. We work with them extensively, but we're not limited to Madison. We work with other companies in the US, in Europe, in Asia. And so we are convinced that and in fact, we've shown it, we can take a very complicated experiment out of your lab and reproduce your work with the same rigor that you would expect. And then more importantly, we can expand that to other compounds that we make. And that's just really an efficient way to do that work. It doesn't bog down your lab with trying to do repetitive assays that your graduate students and postdocs probably just don't want to do. So it's a great fit, and it's worked very well. So how the projects typically work, once we've met you and you've decided to move forward with us, we would ask you to present your work to our scientific advisory board. We have 2 meetings a year. They all fly into Madison. They review our ongoing projects and then talk to the professors about everything they want to talk about. It's really 2 great days of science, really fun. And so we ask you to put together a 1-hour presentation. We provide a template so you can answer some questions that the SAB will always have. But really, you're the expert. You present the data the way you want, the way you like. We have an open question session and then a closed session with SAB, and they give a recommendation. And then, so ideally at that point, your project comes in. Sometimes at stage 0 because the SAB always has questions. And if it's something that we want to answer before we take it into our portfolio, we will do that. That's usually just molecular biology. What happens when you knock out your protein into or out of a specific disease cell line? Often that has already been answered. But if we need to answer it in stage 0, we will pay the professor to do it in their lab. If they don't have that interest in doing that, we will just do it at a CRO. But assuming that data all lines up with your original hypothesis, then move it into stage 1. Or if that data has already been generated, then we start in stage 1. And at that point, WARF Therapeutics is all in. We are committed to find a drug for your disease or prove to ourselves and you that that's just not likely to happen. At that point, we work entirely in parallel with your lab. We work through generate all the data at CROs, pay for it all, of course, share it with you. We ideally would like the professor to act as an advisor, review all our data because you're the expert. But you don't have to do anything in your lab at that point. You, of course, can, and we like that when you do, but there's no obligation. At that point, we are driving it forward through our CROs. And at the end of the day, maybe if we see a drug, you'll see some revenue from that. So I'd really like to convince you to bring your projects. At worst, even if it was outright rejected, you get incredible information from our scientific advisory board. These guys are really brilliant, and they will give you insight that you probably haven't thought about being in an academic setting. That's an unlikely scenario. We accept most projects. At the other end, of course, is the lottery ticket to win a drug, getting a drug, becoming famous, et cetera. Of course, that's a long shot. Most projects don't do that, but it happens. Ask Hector DeLuca. The most likely outcome is a multi-year collaboration where we work together for years, establishing great science around a project you're passionate about. We generate science that you probably would have no other way of generating, certainly not through NIH funding. So I can't promise you we'll take your project on, but most projects that go through our SAB meetings get funded at least at stage 0. And more importantly, I think most people that bring us a project bring us another project. They bring us multiple projects because I think they see the value in what we do, and they really enjoy working with us because we really enjoy working with them. So I encourage you to bring us a project. This slide just shows our current portfolio. This is a highly dynamic slide. It changes a lot, and it could easily be highly out of date by the time you watch this presentation. But I would encourage you to go to our website where we keep an up-to-date version of this. I just wanted to show it to you to convince you that we are capable of driving projects out of stage 0 and onward. So at the time of recording, we had 12 ongoing projects. Half of them had been driven beyond stage 0, and one of them we had identified a development candidate that we were actively trying to license at the time of this recording. Our expertise is certainly small molecules and protein degraders. We do that a lot, and I think we're very good at it. But don't let that stop you from bringing a project to us if you think that your disease or biological process wouldn't easily be drugged that way. We work on peptides and antibodies conjugated to drugs, and even cellular therapies. We've got projects in all of those realms. And if our scientific advisory board thinks we should be working on your project, we will find a way to do it. If we don't have the expertise, we will get that expertise. So thank you so much for watching, and we really hope to hear from you. If you have any questions, reach out to any one of us, myself, anybody at WARF Therapeutics, or anybody at WARF who will put you in contact with us.